Hey everyone, Dr. D here, and in this video we are covering Chapter 15 from our Talaro's Foundations in Microbiology 12th edition by Barry Chess. Let's go ahead and get started.
Dr. D, 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 Dr. D. Explain stuff.
All right, let's go ahead and get started with Chapter 15. Remember, this is the other branch of the immune system. Here we're talking about adaptive or specific immunity, as well as immunization. And do you remember I mentioned last time that adaptive or specific immunity has a memory, and it involves mainly the B cells and the T cells, also known as the B and T lymphocytes. So let's go ahead and get started here. This is also known as your third line of defense. Remember, we already spoke about the first two lines of defenses in chapter 14, the previous chapter. This is the third line of defense. Adaptive or specific or even acquired immunity. Those are three different terms that refer to the same thing. Adaptive, acquired, and specific immunity all mean the same thing. Just like the previous chapter was innate, non-specific, and natural immunity. That also has three terms to it that mean the same thing. An important term to learn for this chapter is antigens. Antigens are any molecule that can stimulate an immune response. And that response in the third line is by the B and the T cells. So think of them as substances that B and T cells can identify and recognize. Foreign antigens are molecules found on germs or anything else that doesn't belong in your body. And self-antigens are your own molecules, your own cells, your own proteins. If you're wondering what sets the adaptive immune system apart from the previous chapter, the innate immune system, it's really specificity and memory. So we're going to talk about both of these. Like, what does it mean? For now, understand that each B cell is specific for a particular antigen, and each B cell is specific for a particular antigen as well. And you should know that the B cells and the T cells can form into what are known as memory cells. And these are cells that their job is to just linger in your body and remember a previous antigen insult, like a previous viral infection or a previous bacterial infection. They form memory cells, which are essentially cells that are programmed to recall their first encounter. That way, the second time you are infected with the same exact germ, you're going to have a much more quick and robust immune response. And that can actually lead to what's called immunization, right? So if you're immune to a virus, usually what happens is you catch, let's say, a particular strain of the flu, right? You catch that strain of the flu, and yeah, it's pretty bad that time, but your memory cells form, B cells, T cells, they form memory cells. And the next time, if that same strain of flu were to get into your system, well, they're right on the job, and you might not even feel sick at all. You might fight it off so fast that you never became sick, right? So that's kind of the job of the third line, this adaptive line of defense. It's the whole reason vaccines work. It's the whole reason why you can't catch the same viral infection twice, if it's like the flu or a cold or something like that, or the same strain of COVID twice, it's like I said, it's the reason why you become immune. And with each insult, with each viral infection or infection you have, your immune system builds more and more memory. And those are based on those antibodies and things like that. So that's what this whole chapter is about. And do you remember from chapter 14 when we mentioned that the lymphocytes, right, the B lymphocytes and the T lymphocytes, I'm just going to call them B cells and T cells. Do you remember that I mentioned They are produced by hematopoiesis in the bone marrow, right? From those hematopoietic stem cells. Those stem cells differentiate into B cells and T cells. The B cells mature in the bone marrow, and the T cells mature where? Can you remember and can you beat Wicket? That's right, as always, Wicket. T cells mature in the thymus. B cells mature in the bone marrow. And I'm going to tell you what that means to mature, because these cells need to mature. And then after that, they need to become activated. So B cells and T cells are really interesting little cells. And do you remember how we mentioned in chapter 14 that B cells and T cells then migrate to those lymphoid tissues, such as the lymph nodes you see here on this slide? After those cells mature, they travel to lymph nodes, they travel to your spleen, they can travel to the salt or the galt, right? They hang out in the lymphatics. They hang out in lymphoid tissues, and their job is to hang out and just wait until they happen to stumble across an antigen, remember, a foreign molecule that they recognize. And once they do, that's really what triggers them to activate. And I'm going to talk about how do B cells get activated, right? How do T cells get activated? Because until they get activated, they're just kind of sitting there. And remember, each B cell recognizes a different antigen, and each T cell recognizes a different antigen. That's part of what specificity means in this chapter. Specifically, we're going to talk about 3 types of cells in the adaptive or specific immune response. We're going to talk about these right here, these B cells. We're going to talk about these are called the helper T cells or T helper cells. And we're going to talk about cytotoxic T cells. Those are the 3 types of lymphocytes we're going to focus on. Now, in order to understand how the B cells and the T cells work, you really have to understand this concept we've been talking about, specificity. Unlike macrophages, dendritic cells, or neutrophils, B cells and T cells have a very, very, very specific receptor on their surface. It's almost like your house key, okay? You see my house key? My house key opens up my house, right? And your house key opens up your house, right? Everyone's got a house key, right? Everyone has a house key. And hopefully no one has the same teeth on their house key because everyone's got a different house and everyone's got a different lock. So hopefully my key is not the same as your key. So what you should think about is, imagine if each and every one of us who's watching this video has a key, and we're all B cells, right? If we're all B cells and we all have this key, that means we all have a unique looking key that opens a specific lock, right? My key does not open your lock. Your key doesn't open my lock. But every B cell has a key, right? Every B cell has a key. And that key sticks off the membrane like this, like it just sticks out of the membrane. It's called the B cell receptor. It's called the B cell receptor. So think of every single B cell in your body has its own specific key with its own specific teeth, right? And that means each B cell recognizes a different antigen. Doesn't that make sense? So for instance, if you come down with COVID, if you catch COVID, for instance, there might be maybe just a tiny handful of cells in your whole body that have the right key, the right B cell receptor to recognize that specific virus, right? That specific COVID virus. It might recognize the spike protein on the surface of the virus. It might recognize the capsomere, the capsid proteins on the surface of the virus. It might recognize, well, it's an enveloped virus, so it might recognize something else on the enveloped virus. Who knows? But it recognizes the virus. So let me ask you this. Let's see if you've been paying attention, and let's see if you can beat Wicket. Can any B cell in your body recognize COVID? That's right. Wicket's always right, right? No, each B cell has a unique pattern on its B cell receptor, right? And that means that each B cell is going to become activated by binding to a specific antigen, right? And for the most part, no two B cells are going to have the same B cell receptor, the same receptor on their surface. So no two B cells are going to recognize the exact same spot on the antigen. Does that make sense? So think about it that way. And T cells are the same way. T cells have the T cell receptor on their surface, and it's the same concept. Each T cell has specific patterns on its T cell receptor called variable regions, and that recognizes a specific antigen. So I just want you to make a note of that. I want you to pay attention to that. No two T cells recognize the same thing. No two B cells recognize the same thing. And that's because they each should have a special, unique receptor on their surface. B cells have a B cell receptor on their surface, which is also called an antibody. I'll bet you've heard of antibodies before. The B cell receptor is an antibody. Okay, but I want you to pay attention to this. The T cell receptor, even though it's very similar, it has teeth, it's smaller and it's not called an antibody. Okay, T cell receptors are not antibodies. They're just called T cell receptors, whereas B cell receptors are antibodies. And each B cell receptor, each B cell has a unique key, and each T cell has a unique key. I mean, not just one key. I mean, the B cell's coded with this B cell receptor, but all of the B cell receptors on one cell are identical. Does that make sense? So imagine if I had 1,000 of this exact key and they're all sticking out of my body, right? Does that make sense? And then you're walking around and you have your house key, 1,000 copies of your house key sticking out of your body. Imagine that's what B cells are. And this is what the B cell receptor looks like. Remember I said like a key, the B cell receptor has this little receptor that's kind of like a key sticking out of its membrane. This is what it looks like. This is an antibody. This is also known as a B cell receptor. Notice that it looks like a Y, right? It looks like a Y. And it's made up of 4 proteins. You see this here? Look at this. This is one protein. And right here, this is called the light chain, right? The light chain, because it's small protein. The one next to it is the heavy chain. Notice how it's a bigger protein. By the way, the 2 heavy chains on an antibody are identical, right? The 2 heavy chains on a particular antibody are identical, and the 2 light chains are identical as well. And so there's 4 proteins, and you can think of this part here. You see where it says C? You see the parts of the protein where it says C? That's known as the constant region, and it's kind of like your keys, right? Look at your keys. I want to show you something. See my key here? Here you can see that there's this part of the key that's identical. Like all keys have this base part, right? That all keys have this constant region. They have this base part that's the same for every key. The thing that makes the keys different are the teeth. Does that make sense? So you can think of the constant region of an antibody as the part of your key that's the same regardless of the key. So that part doesn't change, right? So this part doesn't change. The constant part is just cookie cutter, right? Whereas look up here, you see V stands for variable. V stands for variable. These essentially make up the teeth. These make up the teeth of your antibody. And that's very, very, very hyper unique, right? And so this variable portion is what's going to bind to the antigen. It's called the antigen binding site. And in fact, I'm going to take you to the board. You're going to see younger Dr. D explain what an antibody looks like at the board. Let's do that real quick. Hey everyone, Dr. D here. In this video I'm going to explain what antibodies look like. Antibodies are proteins. They are actually 4 proteins together as a quaternary structure. You've got 2 identical heavy chains and 2 identical light chains. Okay? 2 heavy chains, 2 light chains. That's a protein, that's a protein, that's a protein, that's a protein. 4 proteins. These 2 are identical. Those 2 are identical. And at the tips, you have what's called the variable region. The variable region. So the heavy chains have a variable region, and the light chains have a variable region. These are called the heavy chains. The longer ones are the heavy chains. Heavy, heavy. And the smaller ones are the light chains, because they're smaller. This blue part's called the variable part. The variable part. And the stem here is called the constant region. So you got the constant region, the variable region, light chains and heavy chain. All right? Now, antibodies are usually found on the surface of B cells. This is a B cell. Antibodies are found on the surface of B cells. That's why sometimes they're called B cell receptors. Now here's what makes one B cell different from another B cell. You see where it says variable region? That's the part of the antibody that can vary. So no 2 B cells have the same variable region. It's the variable region that recognizes antigen, right? So here's a molecule. Here's a molecule called antigen. Antigen. Let's say it's a viral antigen, like a viral protein, okay? You may have millions upon millions of B cells in your body, right? Millions of B cells in your body. But guess what? Maybe only one has the correct variable region of antibody to actually recognize and stick to this antigen. Just like you have different keys. I have a different car key than you. Even if we have the same car. I could have a Toyota, you have the same Toyota, but we have different keys, right? B cells have different keys. So this B cell might recognize this viral antigen, but maybe no other B cell in your body, none of the other millions of B cells in your body recognize that same antigen because they recognize different non-self antigens. Does that make sense? So each B cell is different. Each B cell recognizes a different antigen. So when you get infected by a virus, you got to hope that you have the right B cells. You got to hope that by chance you have the right B cells in your body that are going to recognize that viral antigen. You might not, right? Because it's random chance. Okay, so that's what's interesting. And by the way, each B cell has many antibodies on its surface, right? But they're all clones of one another. I want you to realize that. Each B cell has many antibodies on its surface. So this B cell is covered in antibodies, right? But all of those antibodies are clones. What does that mean? That means every single antibody on the surface of this B cell is a clone. Every single one will recognize that viral antigen. Does that make sense? So each B cell has many, many copies of the exact same key. All right, welcome back from the board. So again, this is the B cell receptor, and the T cell receptor, remember, is a little bit smaller. See, this here is the T cell receptor. Same concept. Look, it has constant regions down here, and it's got that hypervariable region up here. The antigen binding site is up here. And remember, no 2 T cells are going to have the same exact structure here with the antigen binding site, so they all recognize different antigens. By the way, make a note of this. Helper T cells have this structure on their surface as well. It's called CD4, or cluster of differentiation 4. It's an important protein on the surface of helper T cells, and it's also a marker of helper T cells. When scientists want to know what type of blood cell they're working with, if they see this structure on top of the blood cell, they know it's a helper T cell. However, the cytotoxic T cells have this instead. They've got CD8, cluster of differentiation 8. So if you see this on a blood cell, then it's a cytotoxic T cell. If you see this one, it's a helper T cell. Either way, they have the T cell receptor, which again is kind of like an antibody in that it has that hypervariable region and a constant regions. But unlike a B cell receptor, this is not an antibody. And remember, those B cell receptors and T cell receptors have that variable region that recognizes specific antigens, or AGs. And actually, it's a little bit more specific than that. An antigen might be huge. You could have a giant antigen, a giant foreign protein, or a giant foreign molecule. But the specific sites on that molecule that are recognized by antibodies or T cell receptors are called epitopes. See this term here? An epitope. So an epitope is a small molecular group recognized by lymphocytes. So again, imagine if, let me show you something. Imagine if this entire pen is an antigen, then an antibody might stick here, right? An antibody might stick there, but those are two different epitopes. Those are two different epitopes. This antibody sticks to this epitope or this part of the antigen. This other antibody might stick to this epitope, which is a different part of the antigen. So think of the antigen as the foreign molecule and the epitope as the exact binding site of the B cell receptor or the T cell receptor. And so each antigen can have hundreds or thousands, of epitopes on it. So it's those complex macromolecules that are the most immunogenic. This term immunogenic means triggers the immune system, activates the immune system. And remember, that epitope or antigenic determinant fits with the receptor, the T cell receptor or the B cell receptor, the way a key fits a lock. Just that specific, right? Very, very specific. And again, do you remember we talked about B cell and T cell maturation? I told you that B cells mature in the bone marrow, T cells mature in the thymus, and then after they mature, they migrate to those lymphoid organs such as lymph nodes in order to hang out until their little key, their little T cell receptor or B cell receptor actually touches the antigen that it recognizes, or I should say the epitope on the antigen that it recognizes. And then that's when it becomes activated, right? But again, what does it mean for a B cell or a T cell to mature? Let's talk about that. What's happening in the bone marrow or the thymus during the maturation process? So here is what's happening during the maturation process. This process is known as clonal deletion. So what's going on? Okay, during maturation, B cells or T cells, pretend these are either B cells or T cells, are formed. And remember, each get a specific key. That's why these receptors look different. See, each one has a specific key. Each one has a specific variable region on its receptor. If one of these receptors, if one of these keys, or I should say receptors, binds to a self antigen, is that good? Let me know. Like, what are you thinking? Should Should a B cell or a T cell recognize self? That means a self antigen, let's say a protein on your own cells. Can you beat Wicket? What do you think? Should a T cell or a B cell recognize your cells as foreign? Yes or no? That's right, Wicket. Yeah, definitely not. That's not good. Because yeah, if you're making B cells and T cells, and each one's getting a different key, right? And that key is what it's going to recognize and try to attack, right? Well, you don't want that key to recognize your home, right? You don't want it to recognize your own self. In fact, when that happens, that can happen, by the way. It's called an autoimmune disease. Have you ever heard of an autoimmune disorder or an autoimmune disease? That's when your own white blood cells recognize your own cells, your own self antigens as foreign, and they start trying to attack it, right? So they might, attack your lungs or attack your liver, and that's not good, right? Because then you get tissue damage. Okay. So what happens during maturation is any B cell or T cell that recognizes self is eliminated. That means that it undergoes apoptosis, which means programmed cell death. And that's good. You don't want cells forming with keys that recognize self, right? With B cell receptors or T cell receptors that recognize self. You only want B cells and T cells forming that do not recognize self because they could recognize something foreign. Like this one might recognize a protein on the surface of COVID virus. This one might recognize a protein on the surface of E. coli. You never know, right? And then you want only those guys to travel to those lymphoid tissues like the lymph nodes in order to hang out. And by the way, these are known as mature B cells or mature T cells, but they're also at this point, they're called naive B cells or naive T cells. And they're going to remain naive. Naive meaning they're not actively doing anything. They're just hanging out until they recognize the antigen. Like I'm hanging out until the antigen I recognize brushes past and binds, and then I'm activated, right? And so naive B cells and naive T cells are ones that they've matured, but they have not encountered the antigen that their key recognizes. And that leads us to concept check number 1. All lymphocytes have the same receptor specificity. What do you think? Can you beat Wicket? That's exactly right, Wicket. No, they don't, right? This is true for all macrophages. Remember macrophages, neutrophils, dendritic cells? These are cells that they really do have the same specificity because they have those, remember, PRRs, those pattern recognition receptors, and those are pretty cookie cutter, right? But remember, B cells and T cells are anything but cookie cutter. Each one is very specific with the key, the receptor that it has. So the answer is 100% false because each B cell and each T cell is actually specific and different from another B cell or T cell. One more thing about antibodies real quick is that they're also known as immunoglobulins or IGs. So if you ever see this term immunoglobulin or IG for short, that refers to a B cell antibody. Now, before I can tell you how B cells and T cells work, I have to explain this concept here because without understanding this concept, you're not going to understand how the B cells and T cells work. So let's pay attention to this right here. There are proteins on the surface of your nucleated cells called major histocompatibility complexes, or MHCs for short. Again, these are proteins on the surface of your nucleated cells, or surface proteins. These play a central role in the acquired immune system. Without these MHC proteins on the surfaces of your cells, your B cells and T cells wouldn't do anything. The third line of defense wouldn't be able to work. And by the way, In humans, the MHC is actually called the human leukocyte antigen, or HLA, but you don't need to know that for now. Just if you ever see this term, that's what it refers to. It's the human MHC protein. But what I want you to know is this. These MHC proteins are found on your nucleated cell, but there are two classes that I want you to know about. I want you to know about MHC class 1 and MHC class 2 because different types of cells have these. MHC class 1 is found on all of your nucleated cells. That means any cell that has a nucleus, such as your skin cell, your brain cells, your heart cells, all of those have the MHC1 gene, which means they can make the MHC1 surface protein on the surface of the cell. Now, this is what I want you to understand. In addition to MHC1, the macrophages, dendritic cells, and the B cells also have MHC2. Okay, they also have a different surface protein called MHC2. And because of this, these three cells, these three white blood cells, the macrophages, dendritic cells, and B cells, are called the professional antigen presenting cells. So if you ever hear this term professional antigen presenting cells, you should think of the macrophages, dendritic cells, and B cells, and that the APCs for short. In fact, let's go over to the board where younger Dr. D will explain this in more detail. Hey everyone, Dr. D here. In this video, I'm going to quickly explain the difference between MHC1 and MHC2. What you need to know is that your nucleated cells in your body, what does that mean? Any cell with a nucleus. Some of the cells without a nucleus would include your red blood cells, your platelet cells. Okay, we're not talking about those red blood cells or platelet cells, but the nucleated cells of your body, your brain cells, your heart cells, your liver cells, your skin cells. Any nucleated cell has a structure, a protein on the top called MHC I, Major Histocompatibility Complex 1. MHC I. I think I've drawn it before with a arrow at the top. MHC I. MHC I is on the surface of your nucleated cells. This is all cells, including, this is what some students confuse sometimes, even your professional antigen presenting cells, the B cells, the dendritic cells, the macrophages, even those cells have MHC I on their surface. So all your cells have MHC I on your surface. The nucleated cells have MHC I on their surface. All right, whereas only your professional antigen presenting cells, those are your B cells, dendritic cells, and the macrophages. The B cells, dendritic cells, macrophages, these are the cells of your body that, in addition to having MHC I, they have MHC II. Okay? So there you go. That's how you know if a cell has MHC I or MHC II. All of your nucleated cells have MHC I, but only the professional antigen presenting cells, the B cells, the dendritic cells, and the macrophages have both. All right, welcome back from the board. I just want to show you this. This is kind of a depiction of what they look like. The class 1 MHC, right? MHC I is this teal one on the left. And remember, that's found on all of your nucleated cells, while MHC II, which is on the right, this one is found on only those professional antigen presenting cells. Remember, the macrophages, dendritic cells, and the B cells. Now, to explain what these do, because they do something, That's something, by the way, MHC I and MHC II, their job is to present antigen, right? Their job is to present antigen. So I'm going to show you, younger Dr. D is going to tell you what that means right now. Hey everyone, Dr. D here. In this video, I'm going to explain antigen presentation. Antigen presentation in nucleated cells, the cells of your body in general, and antigen presentation on professional antigen presenting cells. Remember, these include the B cells, the dendritic cells, and the macrophages. So let's start with your normal nucleated cells, the nucleated cells of your body. Okay? Remember, those cells have MHC I, major histocompatibility complex 1, on their surface. What is the importance of that? MHC molecules are like trophy cases. MHC molecules can bring antigen from inside of the cell and present it outside. Okay? Now, don't forget, natural killer cells, what they will do, sometimes if cells are sick, they will bring the MHC molecule in. Okay? So the MHC will get internalized. That's what they call it, internalized. Natural killer cells notice the internalization of MHC I and destroy that cell. Okay? But let's talk about what the normal job of MHC I is. Okay? What happens is your nucleated cells are constantly taking a random sampling of proteins and other molecules from inside of the cell, handing those molecules to MHC I and letting MHC I present those antigens on the surface. You see this dot I drew right here? That's some antigen, some protein or some antigen that was inside of the cell but was given to the trophy case, MHC I, so it could present it on the outside of the cell. Now you might be wondering, why? Well, because these are called self-antigens. These are proteins that should exist inside of your cells, and your white blood cells recognize these self-antigens and don't do anything about it. But what if this cell got infected? Let me grab a different color marker. What if this cell got infected by a virus? Okay, the virus made itself, at home inside of the cell. Well, if that virus were to make more of itself, it would be making viral proteins, correct? Now, what if MHC I, instead of grabbing a self-antigen and presenting that on the surface of the cell, what if it grabbed a non-self-antigen, like a viral antigen? So now what is it presenting on the outside of the cell? That's right, a viral antigen. That's not self, okay? Did the cell know that it's presenting something that's non-self? No, but guess what? The white blood cells will recognize that viral antigen as non-self. It'll say, hey, buddy, what are you presenting here? That's not supposed to be inside of you. Do you realize you just presented me with some foreign molecule of, a viral? It doesn't know it's a viral molecule, but it knows it's a foreign molecule. Well, you shouldn't have foreign molecules inside of the cell. We're going to have to destroy you now. Okay? So that's how the immune system reacts. So we're going to get into this. We're going to get into this, but it's the cytotoxic T cells, or the sometimes called TC cells. Those are the T cells, the cytotoxic T cells. These are the T cells that have the specific receptors that can identify non-self antigens and do something and become activated. Does that make sense? So your TC cells or CTL cells, these are your cytotoxic T cells. That's their job. Their job is to find nucleated cells. So your skin cell, your brain cell, your heart cell, nucleated cells, scan for their MHC I and look at what antigen is on it. If a self antigen is on it, good for you. It moves on. Everything checks out. If a non-self antigen, like some viral antigen, is presented, it will recognize that as non-self and it will destroy that cell. It will release granzymes and other molecules that promote apoptosis, program cell death in this nucleated cell. Okay, so that's the beauty of it. Now, of course, these TC cells don't. They're going to need extra help with other signals from other white blood cells in order to do that. They don't just get activated right there and then and destroy. Usually, they need a second handshake. But we'll get to that later on. But that's fundamentally what the cytotoxic T cells are doing, if that makes any sense. So now let's talk about professional antigen presenting cells. Professional antigen presenting cells, these include the B cells, the dendritic cells, and the macrophages. What they present on their surface is MHC II. There you go. This is MHC II. And the blue dot I drew is a foreign molecule, all right? So MHC II presents foreign molecules. And MHC II is not recognized by cytotoxic T cells or TC cells. MHC II is recognized by a different T cell. Okay. In this case, it's called a TH cell or a helper. A helper T cell. Helper T cells have specific T cell receptors that recognize foreign antigen on MHC II. So your B cells, your dendritic cells, and macrophages present foreign antigen on MHC II, and that's recognized by T helper cells. So there you go. The difference between MHC I on your nucleated cells and MHC II on the professional antigen presenting cells. MHC I signals to cytotoxic T cells or TC cells. MHC II signals to helper cells, helper T cells. And remember, only the cytotoxic T cell with the correct T cell and receptor could recognize that antigen. So it might be 1 in a million, it might be 1 in 10 million cell that can recognize that specific antigen. Same thing with the helper T cell. Only the helper T cell with the very, very unique T cell receptor is going to recognize that antigen. It might be 1 in a million helper T cells that recognize that one antigen, or even more rare, okay? But once that recognition happens, that leads to activation of the TC cell, that leads to activation of the helper T cell, and that spurs the process, right, of adaptive immune system, or the specific immune system. Hey everyone, Dr. D here. In this video, I'm going to show you T helper activation, or sometimes called helper T cell activation, followed by B cell activation. Let's solve this mystery together, shall we? So, remember that your professional antigen presenting cells have a molecule on their surface called MHC II for major histocompatibility complex 2. And on that, they present foreign antigen. So this blue protein, for example, would be a viral protein. So for example, let me give you an example. If this was a macrophage, professional antigen presenting cell, it may have brought a viral molecule inside, a viral, I should say a virion, a virus inside. Once it brings the virus inside, or the virus infects the cell, the virus will be broken down into antigen. those antigen will be placed onto MHC2, MHC2, and MHC2 will go to the surface of the cell, presenting that antigen to the world. Okay? So again, a virus was either phagocytosed by the macrophage or even infected the macrophage, it doesn't matter, got into the macrophage somehow. That virus gets chopped up. Part of it, each part is called the antigen, part of it gets conjugated with MHC2, and MHC2 presents that antigen. That antigen was a part of that virus, presents it to the outside, right? Because that's what MHC2 is. It's like a trophy case. It presents things to the outside world. And what recognizes the antigen presented by MHC2 on your professional antigen presenting cells? That's right, your T helper or helper T cells. It's the helper T cells that recognize, that have, let me show you here, they have a T cell receptor, a T cell receptor that recognizes foreign antigen specifically presented by your professional antigen presenting cells by MHC2, okay? Now, please be aware that not all helper T cells can recognize the same antigen. In fact, it's quite the opposite. Helper T cells each identify one specific antigen, one random non-self antigen. So it might be 1 in a million or 1 in 10 million or more. One of those T helpers in your body might recognize that antigen. It's like having car keys, right? Everyone could take out their car keys and everyone's car key is going to be shaped a little bit differently. Well, then each of us would be an example of a T helper cell. I have a different car key. You have a different car key. Every T helper cell recognizes a different antigen. So only the T helper cell with the precise T cell receptor that could recognize this foreign antigen is going to bind and recognize this foreign antigen on this macrophage. So what happens right here, this T helper cell goes from a naive T helper cell to a halfway, 1/2 activated cell. So now the T cell, the T helper cell, is no longer naive. Okay? Is no longer naive. It is now halfway activated. Okay? They call that sometimes the first handshake. All right? So, how does the T helper cell become fully activated, you might wonder? Well, that's where the next part of the story comes in. This is called B cell activation. B cell activation. So you've got B cells. Here's a B cell. And what's on the surface of a B cell? Do these look familiar? B cells have antibodies on their surface. Antibodies. Sometimes antibodies are called B cell receptors. Makes sense. But antibodies recognize specific antigens. Just like I said, the T cell receptor is like your car keys. It only recognizes one car, right? Antibodies are the same way. The tip here is a variable tip, right? So every B cell has a different tip, has a different variable portion at its tip. So every B cell recognizes a very, very specific antigen. Just like every T cell recognizes a very, very specific antigen with its T cell receptor. B cells have antibodies called B cell receptors. They recognize something specific too. So let's just say this B cell has the specific antibody that recognizes this very same virus. You remember this virus from back here? Let's say that virus is floating around in your body. That virus sticks to the antibody, right? Because that virus is floating around in your body. You have an infection, right? that virus sticks to the antibody on the B cell receptor. They adhere. What does the B cell do? The B cell's job is to take this antibody virus conjugation, bring this inside. So you've now internalized the virus. And what else do? You chop it up into parts, right? You chop it up. And guess what? B cells are, just like macrophages, B cells are professional antigen presenting cells. So they also have inside of them MHC II. So they'll take this viral protein, conjugate it with MHC II, and guess what they'll do? They will present on MHC II, look, this is MHC II, they will present that antigen from that virus. And guess what? It has to be the exact same antigen as this macrophage was presenting. You see, these two have to be identical. These two have to be identical. The virus antigen that's presented by this macrophage has to match the virus antigen presented by this B cell to be recognized by the same helper T cell. This helper T cell had the right key, right? The right T cell receptor to recognize this viral antigen presented on this macrophage. This helper T cell was halfway activated. Well, guess what? It's about to become fully activated. You know why? Because with its T cell receptor, it's going to recognize, you see, it's going to recognize the same antigen presented by B cells. That's the second handshake it needed. So the helper T cell proliferates and differentiates. So what does that mean? It's going to copy itself because now it realizes it has a very important key. It has a very important T cell receptor. It has the T cell receptor that can recognize this intruder, this virus. So it's going to make many, many, many copies of itself. Okay? And it's going to make effector helper cells. And it's going to make memory helper T cells. So what it does, once it gets that second handshake, it multiplies, it differentiates. Some of those cells become memory cells so that they linger in the body to remember the insult for next time, so that you can become immune to that virus for the next time. Some of those cells hang out and take care of business. And by take care of business, I mean release cytokines and release other hormones that are required to do something about this viral infection now. Okay? What else? By the way, I told you I would tell you about B cell activation, right? When the B cell recognized the virus with its antibody, that binding and internalization was 1/2 activation of the B cell. So the B cell was 1/2 activated. Its second handshake occurred when this semi-activated helper T cell recognized the antigen on the B cell. Now the B cell becomes activated. And what do B cells do when they're activated? That's right, they proliferate and differentiate. They form these massive cells called plasma cells. I'll tell you what those do in a minute. And they form other cells called memory B cells. You know what the memory B cells go do, right? They linger in the body to remember the insult for next time so that you are effectively immune to this virus the next time. This is how you get acquired immunity. What does the plasma cell do? Do you remember the antibodies on the surface of these B cells? Do you remember all these antibody on the surface of these B cells? Well, it was a B cell receptor stuck to the surface of that B cell. Well, now guess what? The plasma cell produces tens of thousands of these antibodies per minute, and it's releasing those antibodies into the bloodstream. So look, the antibodies that were on the surface of this B cell are now secreted into the bloodstream. Does that make sense? So you've got antibodies released into the bloodstream. It's like if I went to Lowe's and I made a bunch of copies of my key and I just made it rain with my key, right? I just threw a ton of my key out, right? Why? Because I know my key recognizes this virus. And that's another term we're going to learn is called opsonization. If let's say this antibody is flowing around in my blood, what if it encounters the virus? What's going to happen? Once that antibody encounters the virus, it's going to stick to the virus. And that's called opsonization. Opsonization is when a foreign molecule, foreign antigen, is recognized by a molecule, such as an antibody. Antibody attaches to foreign molecule, the virus, right? Virus. And then macrophages recognize the antibody. So basically, macrophages recognize the antibody on the virus. Macrophages will gobble up the antibody-virus complex. All right, I know that's a lot. Replay this video, make it clear. This is how helper T cells are activated in general. This is how B cells are activated in general. And this is how opsonization works and how this feedback loop works. All right, this is how adaptive immunity works in your body. This is how, once you've been infected by a particular virus, you are immune to that strain of that virus in the future, because the memory cells linger in your body for years, decades sometimes, and they remember that virus. So the next time that virus is in your cells, the memory cells are quick to act. They're in your tissues inside of your body. They make the antibodies right away, and they take care of business right away. All right? So that's how it works. And again, please understand that this is a really, really watered down version of how this activation works. And immune response is very, very, very intricate, and there's much more to it than just this, but I just wanted to give you a really, really broad perspective. All right, welcome back. I think at this point you have an understanding, right, of what the cytotoxic T cells do, the B cells, and the helper T cells. So hopefully it makes sense. Again, when a B cell becomes fully activated, it proliferates and differentiates, right? The B cell can become a memory B cell to remember that particular antigen for future infections. This is what builds your immunity. And then those plasma cells form. Remember, the plasma cells are essentially antibody factories. Imagine, okay, my analogy is this. Imagine taking your key, your special house key, going to Lowe's or Home Depot and just making thousands and thousands of copies of this exact key and then making it rain, right? Like pouring them into the bloodstream, essentially. That's what a plasma cell does. The plasma cell, look at this, it just releases thousands and thousands and thousands of that exact key. So instead of it being a surface receptor, right? The B cell receptor was on the surface of the naive B cell. Once it becomes a plasma cell, it just spits them into the blood, right? And that's what happened. That's why you have antibodies in your blood, see? And then those antibodies float around in the blood. And if they ever come into contact, if those antibodies ever come into contact with that particular antigen that they recognize, lock and key style, they will bind to it, right? And then this is called opsonization. This term is called opsonization. When an antibody binds to an antigen, that is known as opsonization. And that means that that antigen, see this green dot right here? That antigen is now marked for clearing by the phagocytic white blood cells. Do you guys remember the neutrophils, the macrophages, and the dendritic cells? Yeah, they're on the hunt for this guy. they're programmed to recognize any antigen bound to an antibody and immediately phagocytose that. Does that make sense? So opsonization refers to binding to or coding an antigen such that it makes it much more likely to be phagocytosed, right, by the white blood cells of the immune system. And again, this is why you have antibodies floating around in your blood. It's thanks to those plasma cells or activated B cells. And meanwhile, these little memory cells are lingering in your lymphoid tissues like lymph nodes and spleen, etc. And their job is to linger for years until that particular antigen shows its face again, right? And then immediately, these memory B cells can spring into action. They don't need that second handshake from a helper T cell in order to become activated and proliferate and differentiate into plasma cells and start this process again. And so oftentimes, if you catch the same cold again, or catch the same flu virus again, those memory cells activate so quickly that you'll never even feel sick. And do you remember the purpose of the helper T cells? The helper T cells become activated when they recognize antigen presented by an antigen presenting cell, an APC, a professional antigen presenting cell. Here, they recognize antigen on MHC II. And when the helper T cell is activated, it also proliferates and differentiates into memory helper T cells as well as effector helper T cells. And do you remember the effector helper T cells help fully activate B cells? And then do you remember I also told you about the cytotoxic T cells? The cytotoxic T cells recognize foreign antigen on the surface of nucleated cells presented by MHC I, right? MHC I. Yeah, see this on MHC I, if there's foreign antigen on MHC I on any nucleated cell, including a macrophage, that activates the CD8 positive cytotoxic T cell, which will then differentiate and proliferate into memory CD8 T cells, as well as effector activated CD8 T cells, right? Cytotoxic T cells, which will then track down any self cells, right? Any of your cells that display the same antigen, present the same antigen on its MHC1. And do you remember they can then release these enzymes called granzymes, as well as these perforants, which perforate the membrane of your cell, and the granzymes trigger apoptosis, program cell death, and that leads to the destruction of the infected cell. And hopefully at this point you have a good global understanding of what, again, the helper T cells do. Those are the CD4s. The cytotoxic T cells do. Those are the CD8s. And what the B cells do, right? And now you have the privilege of understanding how our immune system functions at its core, right? Obviously, it's much, much, much more complex than that. But that's the fundamental understanding that you need to understand how the immune system works. Isn't that neat? All right, next, I'm going to explain how the natural killer cells work. These cells are technically part of the innate immune system or innate immune response. Not adaptive, but they are important cells in the immune system. So let's go back to the board with younger Dr. D and have him explain to us how the natural killer cells work. So recall that your nucleated cells in your body, your nucleated cells like a skin cell, a heart cell, a liver cell, they have proteins on the surface, and specifically they have a protein called MHC I, Major Histocompatibility Complex 1 protein. And normal nucleated cells have this MHC I as well as some ubiquitous protein on the surface. And that is basically their identification card. When a natural killer cell comes by and scans, it scans your cells. It needs to see that ubiquitous protein, and it needs to find the MHC I molecule as well, so that it knows that this cell checks out, it is self, it is healthy, and it belongs in your system. So the natural killer cell checks for the ubiquitous protein, it checks for MHC I, and then it goes about its merry way. Now, here's the issue. When a cell is sick, and what do I mean by sick? If a cell is infected by a parasite or a virus, if the cell is becoming transformed, which means becoming cancerous, that cell sometimes recognizes that defect, and it will, in response, it will internalize its MHC I. So what does that mean? It will bring the MHC I in. Okay, so the MHC I molecule is no longer at the surface of the cell. Natural killer cells don't like that. Natural killer cells will find the ubiquitous protein, but then they won't find the MHC I at the surface of the nucleated cell. This triggers the natural killer cells to release granzymes, release molecules which will destroy, induce apoptosis in that nucleated cell. So that's how natural killer cells work. Healthy cells present this ubiquitous protein as well as MHC I. Unhealthy cells can internalize MHC I, and natural killer cells have a job. They are the white blood cells that recognized internalized MHC I and destroy those cells. Why did they destroy those cells? Because they know that the only reason to internalize MHC I is because something's wrong. All right, welcome back from the board. I just remembered it's time for break time with Gizmo and Wicket. So let's do that. Come back strong for more. All right, welcome back from break time with Gizmo and Wicket. Again, we just heard younger Dr. D talking about natural killer cells. And here on this image, I like this image. It's showing you how a natural killer cell works. You can see here that this cell, it says foreign cell, but it does not have to be a foreign cell. Remember, this is a cell that has internalized its MHC I receptors, and that's noticed by a natural killer cell. Natural killer cells release perforins, which poke holes in the membrane of the cell, and then granzymes, which trigger apoptosis, which means programmed cell death, and then the cell dies. Once the cell is destroyed, the cell will get phagocytosed by a phagocytic white blood cell, such as a macrophage, for cleanup. And in fact, this is a neat little picture. It's an actual image with a scanning electron microscope of a actual natural killer cell. Isn't that neat? All right, now let me tell you a little bit more about the immunoglobulins. Remember, these are IgGs or antibodies. You can think of them as the B cell receptors, the antibodies. Did you know that the B cells have about 150 different genes that code for the variable region on the light chains, 250 genes for the variable and diversity regions on the heavy chains, and only a small number of genes for the constant regions and the joining regions of the final molecule. So what does this all mean? I want you to remember V, D, and J. So just remember V, D, J. These are genes that make up an antibody, right? These are the genes that make up antibodies. And now, in addition to V, D, J, you have the constant regions. Remember the part of the key that's the same? So that's there as well. That's another gene as well. So you got V, D, J, C, and there are several V's, several D's, several J's, and several C's. So here you can see what I'm talking about. with the heavy chain. See here on the left where my mouse is, the heavy chain is made up of one of 65 different variable genes, one of 27 different diversity genes, one of 6 different joining genes, and one of 5 different constant genes. So when a B cell is being formed, it picks one of the variable regions, it picks one of the diversity regions, it picks one of the joining regions, and it picks a constant region. And that's what gives rise to the protein. And that's one of the reasons why there's so much diversity inside of antibodies. And then on the right, you can see, again, the light chains, same thing with the light chains. A light chain will get one of 82 different variable genes, one of 10 different joining genes, and only a single constant gene to choose from. And so when you assemble those genes, when they become proteins and you assemble those proteins, remember, you get this quaternary structure of 4 proteins, and these are each highly, highly unique. By the way, after you've selected the variable genes, there's also what's known as somatic mutation, extra mutations that happen in the variable region, making them even more unique. And that is how an antibody is made. In case you were wondering, like, how does every antibody have a unique key? You know, how is every single antibody different? You know, what's responsible for that? Well, by selecting a random gene, right? Random variable gene, a random diversity gene, a random joining gene, and a constant gene, you can add diversity, right? You add diversity to the shape of the antibody. And remember, once you've picked those genes, you can even mutate them further, right? They get somatic mutations, which means extra mutations in that gene for more antibody specificities. And by the way, a lot of cellular signaling is happening during this immune response. There are cell signals called cytokines, including interleukin 1 and interleukin 2 that are important to understand. So interleukin 1 is a cytokine. And remember what cytokines are. Again, they are signaling proteins that are secreted that signal to other cells, right? So for instance, the professional antigen presenting cells will secrete interleukin 1 in order to activate helper T cells. And then in turn, the helper T cell will secrete interleukin 2 in order to activate those B cells and some T cells as well. So you see, there's a lot of intercellular signaling going on via these cytokines. And by the way, I just wanted to point something out. Your textbook may refer to B cell immunity as humoral immunity and T cell immunity as cellular immunity. The reason is that B cells end up releasing antibodies into the humor, right, into the fluids of your blood, whereas the T cells are always interacting with the cells in your body. Does that make sense? So that's just a clarifying note in case you're wondering, why are they referring to T cells as cellular immunity and B cells as humoral? That's why. By the way, we're at concept check number 5. Let's try to do this one together. The lymphocyte responsible for recognizing and destroying cancerous cells is it the antigen presenting cell, the B cell, the cytotoxic T cell, or the helper T cell? Can you be Wicket? That's right, Wicket, the cytotoxic T cell. And by the way, yes, another good answer would be a natural killer cell. You know why? Because sometimes a cancer cell will end up internalizing those MHC I proteins from the surface. And remember, that's what triggers a natural killer cell to take action. So another possible answer would have been the natural killer cells. But remember, That's part of the innate immune response. Now, again, this slide should look familiar. This is called B cell activation. Let's see if we can remember how B cells get activated. Remember, this here is a B cell, and B cells have those antibodies that look like little Y's. See, these are antibodies on the surface, also known as the B cell receptor. The B cells also have these MHC IIs as well as MHC Is on their surface. And look what's happened. This is the 1 in a million event. Look, a microbe, looks like a virus. This looks like an enveloped virus, is being recognized by this B cell. Remember, that's a 1 in a million thing. Not any B cell could have recognized the particular antigens or epitopes on the surface of this virus. So this is a very important B cell. This B cell bound to that microbe. And do you remember what that triggers? That triggers phagocytosis. So the B cell will phagocytose the virus and bring it in as in a phagosome, which it will then fuse with a lysosome in order to, remember, digest those macromolecules, break down those proteins into smaller fragments. And then these fragments can serve as antigens. Do you remember this? The B cell is also a professional antigen presenting cell. So that means that it can take these little fragments of that virus and use them as antigens, place them on, remember, place them on MHC II, presenting them to helper T cells. Remember, this needs to be an activated helper T cell. This helper T cell is already activated. The helper T cell will then recognize it with its T cell receptor. And remember, this is also a 1 in a million event. Remember that? This helper T cell, not any helper T cell would recognize this foreign antigen presented by this B cell. Only the right one with the right T cell receptor key, right? Isn't that interesting? So this helper T cell will then release those interleukins I told you about in order to fully activate. Remember the second handshake, fully activate this B cell. And you can see that more closely here. The B cell was presenting that foreign viral antigen on MHC II. The helper T cell recognized it. Remember, helper T cells are CD4 positive cells. Then the B cell becomes what? Remember? The B cell becomes activated, which means it will differentiate and proliferate. And look at this. Some of them differentiate into memory B cells, whose job it is to go and remember, right? Those memory cells with the same specificity remain in the lymphatic circulation. They will live in your lymph nodes. They will live in your spleen. They will live in the tissues. And remember for next time, maybe 2 years from now, okay? And then remember, the activated B cells also differentiate into plasma cells. Plasma cells, which are these big cells that just start, making it rain, this antibodies, right? So the antibodies that used to be on the surface of the B cell are now being just strewn about into the blood, and they travel around into the blood. And, I'm going to explain what IgM antibodies are in a minute, but you can have different versions of this key, different variations of this key. Not variation, by the way, the key, the antibody that's being released by these plasma cells all have the same teeth, but they can have different constant regions. Does that make sense? And that's what makes for the different classes of antibodies that can be secreted by B cells. All right, so now that we've reviewed B cell activation again, let's talk about why these antibodies are so important. When you make it rain, antibodies, why is that so important? Why do we want antibodies in our blood plasma? Why do we want all these antibodies floating around in the blood? So let's talk about that next. So this slide shows you what antibodies are good for. This is the slide labeled antibody-antigen interactions. And look, there are 6 different things that these antibodies can do for you once they are in your blood, right? Once they're secreted by those plasma cells. So I already told you about this one. Let's start with this one. Let's start with this one. We already talked about this one. Remember this term I wanted you to remember, opsonization. Take a look at this term, opsonization. Do you remember what that means? That antibody is going to stick to the antigen it recognizes. So imagine if there is a virus somewhere in your body and this antibody, recognizes that virus, right? So it's going to stick to that virus and that's going to flag it for phagocytosis. Remember your phagocytic white blood cells like macrophages, dendritic cells, neutrophils, they're going to spot a antibody that's stuck to an antigen very easily, and that is easily phagocytosed and identified. So you see what it says here, opsonization, when coated with antibodies, opsonized, microorganisms or other particles are more readily recognized by phagocytes, which engulf and dispose of them. So you see, yeah, I want a bunch of antibodies in my body, right? So for instance, if my body is making a ton of antibodies against the COVID virus, well then those antibodies are going to float around my blood until they bump into a COVID virus, and then they will opsonize that COVID virus so that my macrophages and other phagocytic white blood cells can gobble them up right away. Isn't that neat? But look, antibodies have more jobs than that. So let's talk about this. Do you remember in the previous chapter 14, we talked about complement proteins? Remember there was 30 different complement proteins in your blood? And one thing I want you to know, remember I briefly mentioned that there are 3 ways to activate complement? Well, one of the ways that those complement proteins get activated is by noticing antibodies attached to an antigen. So if, let's say, antibodies are stuck to a bacterial cell, that's going to activate those complement proteins to come help. Isn't that neat? So it has something to do with complement activation. Something else antibodies can do is a process known as neutralization. So for instance, take a look at this. If antibodies stick to the spike proteins of a virus, well, guess what? Now the virus can't stick to the cell. Isn't that neat? So think about it. Remember, viruses need their little spike proteins in order to stick to and adsorb to your cells. However, if antibodies are sticking to all the spike proteins, well then effectively the virus can't stick to your cells, right? So that's called neutralization. Isn't that neat? Next, antibodies can even serve as antitoxins. Imagine if this is a toxin. This is some sort of venom from a snake, right? This venom is toxic to you. However, if antibodies can stick to that venom, well then maybe they can prevent that venom, that toxin, from actually harming your cells. So antibodies can be used as antitoxins. Another thing antibodies can do is precipitation. This is when antigens much smaller than a cell, these are typically proteins, can be bounded by antibodies, increasing the speed with which they are phagocytized, right? So you've got this foreign small matter in your body. Antibodies bind to that foreign tiny matter, making it big and clumpy, causing it to precipitate, and then that is easily identified by phagocytic white blood cells and cleared out of your body. And then the last thing antibodies can do is a process known as agglutination. This is when because you have two arms and a Y, right? Antibodies have 2 arms. So if one arm sticks to one antigen and the other arm sticks to another antigen, that could bring everything together, and that's called agglutination. And the purpose for agglutination would be to speed up phagocytosis and clearing by those white blood cells. So it's not just opsonization that these antibodies can do. These antibodies have many important roles inside of our body. Now, do you remember when I said that antibodies have 5 different constant region genes? See, look at this. This is the constant region of the antibody. That's the part that, is similar for many different antibodies. And do you remember I said there's 5 different constant genes? That means this part of the antibody can be 5 different things while maintaining the same exact variable key, the actual teeth, right? The important part. So uh, B cells can swap out the constant regions in order to make 5 functional classes or isotopes of immunoglobulins or IGs. For instance, one of those constant regions will give you what's called IgG. Another one will give you IgA. Another one will give you IgM, IgD, IgE, and they look like this. Like IgG is your classic Y-shaped antibody. IgA, it kind of forms this like dimer. IgM forms a pentamer. IgD looks like a Y, but it's different. And IgE is a long Y. Okay. And by the way, all of these have the same, the only difference between these is the base, the constant part, not the important teeth. The teeth can be the same, but the base can switch, So the B cell can switch, for instance, the B cell can switch from IgG and then switch it to IgM. It still recognizes the exact same antigen, but you've changed, the way they kind of interact and the constant part, of the antibody. And you might be wondering, why is that interesting or important to understand? Well, it's actually kind of neat. Let me show you something. In the primary response, this is when a B cell is first activated, right? Imagine a B cell becoming activated, right? And now it's become plasma cells, making it rain those antibodies. Did you know that it takes about 10 to 12 days for those B cells to start making antibodies? And then when they do, look at this. Upon primary response, the first exposure, those B cells will first make IgM and then switch to making IgG, right? So that's kind of neat. Think about that. Look, after a B cell is activated, this is what they secrete first, this IgM, and then after, some days, it'll switch to IgG. And you can see that if you take someone's blood, like let's say they get the COVID vaccine, first a bunch of these pop up and then it switches to these, because you're changing the constant region of the antibody. But let me show you something else that's really neat. The second exposure, remember those memory cells that have to remember. Let's say you catch the same virus next year. In this case, look what happens. You have this huge early response, but it's mainly IgG and to a lesser extent IgM. And look, the first exposure, look how long it took to make antibodies. It took 2 weeks. It took about 2 weeks to make antibodies upon the primary response. But do you remember when I said that the second time you get exposed to the same virus, you're going to have a way more robust response? Because look at this. It only takes a couple of days in order to make a ton of antibodies against that virus the second time. And that's why you'll never even feel sick the second time most of the time. And let's just read what it says down here. It says, the secondary response, a latent period is lacking. That means like you don't have to wait 2 weeks. A latent period is lacking because other memory lymphocytes, those memory B cells, from the earlier response are immediately ready to act. A rapid rise in antibody titer, mainly of IgG, is sustained for several weeks. A smaller amount of IgM is also produced by those naive B cells. Okay, so you can see that this is what happens when you're first exposed to a virus and then when you're exposed to it down the road again, right? You see how much bigger that response is and how much quicker that response is. And that's what's responsible for your immunity. Another important concept to understand is how you get your immunity. There are 4 different ways to obtain immunity. There's active immunity, passive immunity, natural immunity, and artificial immunity. So we're going to talk about all four. Think of active immunity as when a person is challenged with an antigen. So the antigen is inside of your body, and that activates, remember, those B cells or T cells, and that stimulates the production of antibodies, creates memory cells, right? It takes time to develop, but it's lasting. So active immunity, you should make a note that active immunity means the the antigen was in your body, your B cells became activated, and they're making all those antibodies and memory cells. Passive immunity, you should think of it as you are not getting exposed to the antigen. Instead, preformed antibodies are donated to an individual. So, for instance, imagine if antibodies from someone else was injected into your system. That would not create the memory cells. It wouldn't stimulate your B cells to make antibodies. Your B cells are not going to remember anything. It is short term. Natural immunity is acquired as part of normal life experience, and artificial immunity is acquired as a medical procedure. So in the clinic, such as a vaccine. A vaccine is an example of artificial immunity. Whereas, catching the cold would be an example of natural immunity because you'll become immune to that strain of the cold virus. So now we can even combine these terms. So let's talk about how do we combine these terms. Let's talk about this. For instance, here's some examples. Natural active immunity would be, for example, getting an infection. Like, so if you come down with COVID, right? It's natural because it's just part of life, and it's active because it's going to stimulate your B cells to make antibodies and memory cells. Does that make sense? Whereas, let me show you this. Natural passive immunity, this would be the example here would be mother to child. Did you know that during breastfeeding, the baby can ingest antibodies, and those antibodies can help with the immunity of the child? And did you know that there are certain antibodies that can cross from the maternal bloodstream into the placenta, of the child? So antibodies from mom, preformed antibodies from mom, are an example of natural passive immunity. It's a short-term version of immunity, and it's natural because it's through breast milk or through the blood barrier. Now, what is artificial passive immunity? This is called immunotherapy. It's artificial because it has to be done in a clinic. So imagine if you've been bitten by that snake, right? Remember that venomous snake? The snake bit you, and so you're trying to get antibodies injected into your body right away as an anti-venom. Does that make sense? So in that case, it's artificial because it's being done in the clinic. It's passive because your B cells aren't going to, do anything. Your B cells aren't going to play a role. It's those antibodies are going to directly go and stick to that toxin, that venom, and neutralize it. Does that make sense? Artificial active, on the other hand, the best example of that is vaccination. If you get a vaccine, right? So a vaccine, let's say they inject you with the spike protein from COVID, and that challenges your immune system, right? It's artificial because the vaccine was done in the clinic, but it's active because it's going to stimulate your immune system. Remember, those B cells get activated, they form memory cells, and they form plasma cells to form antibodies, and you have that long-term immunity. Does that make sense? So hopefully now you understand those four categories. Now let's talk a little more about vaccine preparation. Some vaccines come from killed cells or inactivated viruses. Some vaccines come from live attenuated cells or viruses. That means cells and viruses that, are functional, but attenuated means no longer able to cause disease. Vaccines can come from antigenic molecules derived from bacteria cells or viruses. So imagine, for instance, instead of injecting you with the whole virus, they just inject you with the spike protein from the virus only. In fact, that's how the COVID vaccine works. And also genetically engineered microbes or microbial agents as well. These are 4 common preps for different vaccines. And obviously, a good vaccine has requirements. It should have a low level of adverse side effects or toxicity and cause no harm. It should protect against exposure to natural wild forms of the pathogen. It should stimulate your B cells and T cells to become activated and form those memory cells and antibodies. It should have long-term lasting effects. It should work with minimal doses or boosters. And definitely it should be inexpensive, have a long shelf life, and be easy to administer. What are some advantages of attenuated vaccines? The organism can multiply and produce an infection but not cause a disease. It's very similar to the natural infection. It can confer long-lasting protection, and it requires usually fewer doses and boosters. Disadvantage of this strategy is that it requires special storage and can be transmitted to other people and can conceivably mutate back to a virulent strain. So these can be kind of risky. So at the top here, you can see what they do with killed cells or inactivated viruses. They take cells or viruses and they radiate them or chemically treat them. And now they're dead or they're denatured, but their antigenicity is retained. So those surface proteins are still there, right? Those surface proteins, those antigens are still present, and that's what's included in the vaccine. The bottom example is an attenuated cell or vaccine. In this case, you take either the cell or the virus and you eliminate its virulence, which means you make it so that it can't actually cause the disease. It's still alive or active. It can still cause an infection. It just can't cause the disease. And then these attenuated viruses or cells are bottled up inside of the vaccine. And again, some vaccines come from parts of a pathogen. So, for instance, you could have a vaccine which is made up of the flagella of a bacteria, or specific proteins from inside the bacteria, or the cell wall of the bacteria, or the outer membrane of the bacteria, right? So these are parts of the bacteria, not the entire bacteria. Same thing with viruses, right? Instead of injecting you with the whole virus to give you that immunity, you could get just a spike protein. So instead, you're just being inoculated with that spike protein for immunity. And then lastly, you can use genetic engineering. You can take a gene from a virus and put that into a plasmid, and then have the plasmid injected into a yeast cell, right? Transfected into a yeast cell. And then that yeast cell will produce that protein that came from that original viral gene. And then you can bottle up that protein. Remember, and this is technically a protein from this virus. In this case, it looks like a spike protein from that original virus, but you used genetic engineering in order to produce that protein from the virus. An example of a genetic engineered vaccine is the Johnson & Johnson COVID vaccine. So let's talk about how that works. So here you can see kind of how that Johnson & Johnson vaccine would work. you're taking the spike gene, that's the gene that codes for the spike protein of COVID. You're taking that spike gene out of COVID and you're placing it inside of a harmless virus. You then inject and that virus, this virus is the vaccine. So when you take the vaccine, you're actually being given this virus. I suppose you could call this an attenuated virus because it can't cause disease. It can only cause infection. So then when this virus gets inside of your system, it infects your cells and it delivers what? Remember, it's delivering that gene for the spike protein. Now your cells undergo transcription and translation and produce COVID's spike protein, and that's triggering your immune system, those B cells to become fully activated and form memory cells and plasma cells and antibodies. And see, so that's how the Johnson & Johnson vaccine works. It's a viral delivery mechanism. It's a form of genetic engineering. And then do you recall that the Pfizer and the Moderna vaccines are what are known as mRNA vaccines? These are collectively known as RNA vaccines, where viral RNA or mRNA is inoculated into the recipient. Again, this is how the COVID vaccines worked from Pfizer and Moderna. So here's how that works. Let me show you how that works. Here is the spike protein gene. That's all you need. All you need from COVID, pretend this is COVID right here. All you need is the spike protein gene. You then place that spike protein gene mRNA, right? Not the gene itself, but the mRNA from the spike protein. You put the mRNA inside of little lipid nanoparticles. That means basically a bubble, right? So you know what lipids are, right? Phospholipids. You form little phospholipid balls with mRNA inside. And remember, that mRNA codes for the COVID spike protein. And then that's the vaccine. You inject people, those little nanoparticles touch your cells, they open up and deliver the mRNA. Your ribosomes translate that into spike proteins from COVID, and then your immune system recognizes that. Remember, those B cells become activated to form memory B cells and antibodies from plasma cells and all that good stuff, right? So this is how the mRNA vaccines work. You're being injected with a nanoparticle, a tiny bit of lipid with mRNA for a spike protein inside. Concept check. Last concept check here. Concept check number 9. If only a portion of a pathogen is being used as the basis of a vaccine, it would be would it be an attenuated vaccine? No, because that's the whole cell or the whole bacteria. Is it an acellular vaccine? Yes, because it's just a portion of a cell, not a whole cell. So I would say B. That's the answer there. The last concept, too, I wanted to bring up before we wrap this up is a concept of herd immunity. And that is that the more people are vaccinated towards a particular disease, the more likely it is that that disease will not spread to someone who cannot be immune. There are people who are immunocompromised. There are people who are on chemotherapy. There are people who just can't become, vaccinated. And you're protecting those people by having more of the population have the immunity. This is known as herd immunity. The more people are immune to a particular disease, the less likely it is to be spread around, and the more you are protecting people who cannot become immune. And these vaccines have done a great job over the past 100 years. You can see here that measles affected more than 500,000 people every single year in the 20th century, and now it's down to 5 people a year in 2021. Pertussis, this is what causes whooping cough, was 200,000 people per year in the 20th century, and now that's down to 1,200 today. Mumps, same thing. Look at the 99% reduction. Rubella, a greater than 99% reduction. Smallpox has been eradicated. Polio has been eradicated. And these are all different diseases that, have been significantly reduced, if not eradicated, thanks to the protective effects of vaccines. And with that, that leads to the end of this long but very interesting chapter. I hope you learned a lot. Let me know if you have any questions, and I'll catch you guys next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D,